Hello guys, now we are still on our revision segments. I've done topic 1 and 2, and today's video is going to be based on cell division and, if time permits, gametogenesis. Now, cell division is the process whereby a cell splits into different parts. If it splits into two parts, it is called what? Binary fission. But if it splits into two or more parts, it's called what? Multi-fission. But in today's video, we are going to be discussing mitosis and meiosis. Okay. Now, in mitosis, two daughter cells are produced. In mitosis, two daughter cells are produced. But in meiosis, four daughter cells are produced, and mitosis occurs in a somatic or growth cell, but meiosis occurs in a reproductive cell. Now, in this cell division, we are going to be considering four stages. We have this gap 1 stage, the S stage, gap 2 stage, and the M stage. All right. The gap 1 is characterized by the growth and preparation of duplication of what? Chromosome. All right. Which means that the growth of chromosome occurs in this what? Gap 1 phase. The gap 2, as this as synthesis of DNA and duplication of what? Chromosome. Duplication of what? Chromosome. Now this gap 2 stage is called what? Preparatory stage. All right. This gap 2 stage prepares itself for what? Mitosis stage. And this fourth stage is what? Mitosis. So these are the four stages that occur during what? Cell division. Now, mitosis, we have IP mats. IP mats. This is what? Interphase. This is prophase. This metaphase. This anaphase. And this what? Telophase. Alright, so these are the stages under what? Mitosis. Let's start with the first one. The interphase is not actually known as a stage. It's not among the stage. That is, it's not to be considered as a stage. Hence, it is known as what? The resting stage of the cell. It is the resting stage of the cell. During the interphase, the chromosomes are not yet visible. The chromosomes are not yet what? Visible. They occur as what? Thread. They occur as what? Thread-like structure called what? Chromatin. During the interphase, the chromosomes are not yet visible. Hence, they occur as what? Thread-like structure called what? Chromatin. But during this prophase, okay, let me draw something, please. I need to draw something here. I have this. It's too big. Now, this is the cell membrane. This is called the nuclear envelope. And this is called the nucleolus. these thread-like structures inside, they're actually called the chromatin. So this stage that's just be this diagram that's just been drawn on the board is called the interphase. You can see now that the chromosomes appear as what? Threads, called what? Chromatin. All right. Now the next stage is what? The prophase, which is, I said the interphase is not among the stages. It's known as what? The resting stage of the cell. It is known to be considered as what? As a phase or as a stage. The next one is what? Prophase. Prophase. Now during prophase, one, the nuclear membrane disintegrates. The nuclear membrane disintegrates. Disintegrates. Yes. Then the nucleolus disappear. The nucleolus disappear. Now in this prophase, the chromosomes don't occur as chromatin anymore. The chromatin will condense to form what? Chromosome. Which means that in this prophase, chromosomes are formed when the chromatin in the initial stage condense. They form what? Chromosome. Okay. Now this chromosome, a pair of chromosome will now attach themselves to at a given point called what? Centromere. Or called, some persons call it what? Centromere. And it's not centromere, it's what? Centromere. Now you can see nuclear membrane disintegrates, nucleolus disappears, chromosome is formed, as well as what? Centromere. Look at it here. You have this. Now you have this is a nuclear envelope. This is centromere. Now this middle here, where the chromosomes are found attached, is called what? This is centriole, sorry. Centrioles. You have the centromere. Okay, so you can see the chromosome has been. Do you have a pair of sister what? Chromatids. All right, this two structure here. Now chromosomes have what? Formed. Okay, now that is a what? Prophase. Nuclear membrane disintegrate, nucleus appear, chromosome or whatever. So the next stage is what? Metaphase. Metaphase. During the metaphase, there is formation of what? Fiber. Fiber is formed during what? Metaphase. During metaphase, fiber is what? Formed. So you have this. I look like this. Now during the metaphase, you see them, they will align themselves on this equator. You see the chromosomes, they will attach themselves on this spindle fiber. So it is during the metaphase that spindle fiber is what? Formed. You can see that they are aligned they align themselves on this equator. This point is definitely centromer. Is the isthmus, spindle fiber. These fibers here, where the chromosomes are found attached. Spindle fiber. All right. So they rearrange themselves on the spindle fiber of what? The equator. That is what? The metaphase. Then during the anaphase, during the anaphase, they will split, and the pair of sister chromatids will move what? Towards the opposite pole, like this. Jesus. You can see they were joined together, but during the anaphase, they will move towards opposite pole. One will go up, and the other one will go down. Look at it. You can see one is going up, and the other one is coming down. So the pair of star chromatids will move towards what? Opposite pole. Opposite pole. One is going up, the other one is coming down. Don't forget, pair of star chromatid, spindle fiber, A star. Alright? So take note of that. Then during the telophase, everything that happens during the prophase is the opposite of what happens in the telophase. Okay, let me put it here, please. I have this. The telophase is like this. It's double of what happens in the prophase. Of course, you have chromosomes like this. You have chromosomes. Same thing is also found here. Now you can see now that the cell membrane has what? That is the cytoplasm has. Please, cytokinesis. Cytokinesis occurs during what? The telophase, which means the splitting of what? The cytoplasm of what? The cells, called what? Cytokinesis. Now, there is formation of what? The nucleolus will be formed. Nucleolus will be formed. The cell membrane is what? Reformed, which means there is what? Appearance of the nucleolus and formation of what? The nuclear membrane. That is opposite of what happens. Please, prophase is known as what? The longest phase. Prophase is known as the longest phase, all of what? These phases. Now, the next one, everything I've just discussed now is what? Mitosis. Now, I said two daughter cells are formed during what? Mitosis, and take note of the characteristics. Opposite of what happens in the prophase is applied to what? Telophase. Cytokinesis occurs in what? Telophase. Now, what are the importance of mitosis, or the significance of mitosis? 1, regeneration. 2, growth. 3, replacement of worn out cells and tissues. We have genetic stability and asexual reproduction. So ask which of these is not a significance of mitosis. They'll give you this. They'll give you genetic stability, replacement, growth, regeneration, and genetic variation. Yeah, genetic variation is not applied to mitosis. That one is meiosis. These are the significance of what? Mitosis, generation, growth, replacement, genetic stability, and what? Asexual reproduction. Now let's now discuss meiosis 1. I told that during meiosis, four daughter cells are what? Produced. Four daughter cells are what? Produced. Now, during meiosis 1, we have prophase 1. Prophase 1 is now divided into 5 stages. We have the leptothene, the zygothene, these markers are becoming bad, the pachatine, diplothene, and diakinase. Okay, so which of these is not a stage under prophase 1? They'll give you leptothene, zygothene, pachatine, diplothene, and whatever. These 5 phases are under prophase 1, and as it is the most important phase. Now, during the leptothene, what happens? The chromatin will condense and become shorter. Under this leptothene, the chromatin will condense and become shortened. Now, the zygothene, during zygothene, the homologous chromosome will pair. Homologous chromosome. There's pairing of homologous chromosomes through synapses. Look at it, SIS, through synapses, okay? Now, resulting to a bivalent entity. When synapses is formed, there's bivalent. There's formation of bivalent. Then during the pachatine, these bivalents become clearly seen. Seen. Please take note of this. It is all of these stages mentioned. The most important one is the pachatine stage, because it is during this stage there is crossing over. It is this stage that is crossing over. Take note of that. Now, it is during the diplothene that the homologous chromosomes they are pulling apart to form they now form what is called chiasmata. The chiasmata. All right. During the diplothene, these homologous chromosomes, they get prepared. They are pulling over, and they form a chiasmata. Chiasmata is that point of It's the point of attachment of non-sister chromatid. They'll ask you a point of attachment of non-sister chromatid is called which of these? A point of attachment of non-sister chromatid is called which of these? It is chiasmata, and it is formed during the diplothene stage. Then the diakinase, during the diakinase, the homologous chromosomes pull completely. They pull apart. They eventually pull apart, and during the diakinase, spinofiber appears. That is, it is formed, appears, but the cell membrane and nucleus disappear, which means that during the diakinase, It's what happens during prophase 1 in mitosis. Okay, it is what happens during prophase in mitosis that occurs in diakinase in prophase what? 1, under meiosis 1. There is disappearance of chromosomes and nucleolus. Okay, take note of that. I said during the diakinase, homologous chromosome they pair completely, they pull apart completely, they divide opposite poles, and which one again? And I said the spindle fiber is formed, nucleolus disappear, okay, nucleolus disappear, and nuclear membrane disintegrates. That is for prophase what? 1. The next one is metaphase 1. Please take note of this stage. Chiasmata is formed during diplotene. Diakinase, nucleolus, and cell membrane or nuclear membrane disappear. There's formation of what? Spindle fiber. Pachatine is the most important one because there's what? A crossing over. Okay, take note of that. Chiasmatas, some during diplotene. Okay, the next one is what? Metaphase what? 1. Now that metaphase 1, I don't want to spend my time on that. Okay, there's a lineup of what? Tetrads in the what? Spindle fiber. There's lineup of what? Tetrads in the spindle fiber. That is metaphase 1. IP max. Okay, metaphase 1. Then during anaphase 1, of course, anaphase 1 is the homologous chromosomes, they attach themselves on the equator. They attach themselves on what? The equator. They attach themselves on the equator. During the telophase 1, they actually get what? Their destination. Now, during this telophase, the chromosomes condense to form chromatin. The chromosomes condense to form what? Chromatin, and the nucleus appear. The cell membrane disintegrate. Please, the chromosomes are under the magnification of light microscope. Yes, I stress light. The chromosomes are under the magnificence of what? The light microscope. Please, the father of microscopy is Anthony von Leeuwenhoek. The father of microscopy is called what? Anthony von Leeuwenhoek. He was the first to discover the microscope that was having magnification power of 270 and above. All right, so it's known as father of what? Microscopy. Now the next one is what? Meiosis what? 2. Meiosis 2 is very similar to what happens in mitosis. It's very similar to mitosis. The only one I'm going to be focusing on is metaphase 2. During metaphase 2, there is formation of what? Kinetopore. Please take note, kinetopore is formed in which of these stages? Metaphase 2, kinetopore is what? Produced. That is metaphase 2, kinetopore is what? Produced. What are the significance of what? Meiosis? 1, sexual reproduction, and 2, we have a genetic variation. I told you that it takes meiosis occurs in a reproductive cell, and there's what? Genetic variation. So at this point, to call it a day, please, the chromosome is under the magnification of what? The light microscope. Which of this stage is kinetopore produced? Metaphase 2. Which of the following stages under prophase 1 is the chiasmata produced? It is what? Diplotene. A point of attachment of non-sister chromatid is called what? Chiasmata. Which of the following is the most important stage under prophase 1? It is what? The Pachatine, because there's what? A crossing over. Please take note of that, and also take note of the fact that everything that happens during prophase is the opposite of what happens during what? Telophase. Cytokinesis occurs in telophase. Thanks for watching, and do have a wonderful day.